please put your hands together for Thomas Pucadyl from IISER Pune.
I'm sure everyone knows what this is. This is a ball of dough. And if I were to ask you to make two balls of this dough, and if I were to ask you to break this, let's say, into two pieces, you would probably go ahead and then rip it off, right? Like this. Now, if I were to get you to do the same thing, but very slowly, then something interesting happens. Okay, you see the dough thinning out in the center, and it tries to not want to undergo breakage, but it can't yield and eventually gives way to the force that you're extending, right? Now, what's remarkable is this process is exactly, or the pathway to this process is exactly how every cell undergoes division. It's a manifestation of the type of material that the dough is made of. And these traits and these properties are also present in membranes that are present to encapsulate cells, encapsulate life around them. So this material property is very unique because it responds to forces, it bends, but at the same time, it doesn't like to break. And this is critical because if with every cell division, if the membrane broke, you would be releasing what's inside the cell outside, and you would be releasing or letting in what's outside to come inside. So you would have an imbalance in the chemistry of the cell, which ultimately leads to cell death. So this is a process which has evolved taking into account the material properties of that envelope around cells. And this process of division, which technically is referred to as membrane fission, is what we study. Fission is obviously not a process that can be managed by a single protein because the scales of this entire phenomenon are quite large. Proteins are very tiny entities, but you're talking about micron scales when you're dealing with cells. So there's a collection, there is an orchestration of a variety of different types of molecules that come and manage division. And what is remarkable here is this offers us avenues of coming up with strategies, therapeutic strategies, because just like cells in our body would like to divide, there are also cells that are of a pathogen origin that also like to divide. And this is quite a bit of excitement about developing new antibiotics. The problem, though, is that these antibiotics quickly run out of steam because the pathogen evolves resistance against them. Instead, you could target some of these processes such that it's practically impossible for a pathogen to develop resistance against several proteins at the same time. So that's the focus area of our research, and I'd be more than happy to tell you a lot more about what we do on a one-on-one. Thank you.